presentation, SDS, the challenges and solutions. So I'm going to be referring a lot to SDS, that's sudden death syndrome. Sudden death syndrome, SDS, is a disease that was first observed in southern Illinois in the late 70s and early 80s. But now we see it just about everywhere in Illinois. It's up and into Canada, North Dakota, just about anywhere now that soybeans are produced. Now this title is a little misleading. While we have plenty of challenges when it comes to sudden death syndrome, I wouldn't necessarily say that we have solutions. There are no silver bullets. When we have years that really favor disease, no matter what we do, we're still going to see some symptoms as far as SDS is concerned. We'll begin the presentation with some acknowledgments. So much of the research that I'm going to be sharing with you here today was done by these researchers at various universities, both in the US and in Canada. But I'd like to particularly point out Dr. Darren Mueller at Iowa State University and Dr. Carl Bradley, who's now at the University of Kentucky. You can see that checkoff dollars are well represented as far as funding is concerned, and these are the various funders of some of the research that I'm going to be presenting here for you today. So let's begin. Symptoms of sudden death syndrome begin as this yellow flecking that starts to show up between the main leaf veins. The flecking then coalesces to develop into interveinal chlorosis, which is just a fancy way of saying yellowing between the leaf veins. The chlorosis then turns into necrosis, or dead tissue, and pretty soon, as the disease progresses, the only tissue on the leaflet that might remain green is that main vein tissue. With time, these leaflets might fall off, leaving the petioles attached to that main stem. Now, it's not every year that we see symptoms this severe, but as you can imagine, those years where we do have leaf drop early on in the growing season, there can be a significant effect on yield. So, as with any soil-borne disease, SDS has a very patchy distribution, and that's because the pathogen is not evenly distributed throughout a field. You'll have areas of higher population densities, like probably here, and areas of lower population densities. This, I can assure you, is not a picture of SDS. There's a lookalike out there. Does anybody know what this might be a picture of?
Brown stem rot.
Brown stem rot, great. So the best way to tell the difference between BSR and SDS is to go ahead and split the stem. You can see that along the stem tissue, right near, especially near the leaf axils, you might see this brown discoloration. If you do, it's most likely brown stem rot. That's where it got its disease name. If you don't, you probably have SDS. Not always, but you probably have SDS. SDS can also cause symptoms of root rot. This isn't really rotting of that main taproot, but it'll be the lateral roots that rot. So if you split the taproot of plants that are showing those foliar symptoms, sometimes you can see brown discoloration in the taproot. This is characteristic of SDS. You don't always see this, but you might see this. Plants that have BSR won't have that brown discoloration in the taproot. Okay. Here's another way to help in diagnosing this disease. So you see this bluish-white fungal growth here. I took this picture last year. There are several plants that were showing symptoms at the research center where I work. So I gently dug up the plants, or the roots, shook off the soil, and I saw this fluffy bluish-white growth. That's actually the fungus that causes sudden death syndrome. You're not always going to see this, but if you do see it, you have SDS. The fungus that causes SDS is called Fusarium virguliforme. So this pathogen is soil-borne. It's the same pathogen that causes SDS in South America, but you may notice that there are 3 other additional Fusarium species that can also cause this disease in South America. Here's the disease cycle. So we used to think that sudden death syndrome was favored only by cool, wet weather at planting time. We now know it's a combination of that, so the cool, wet weather at planting time, but also plenty of rain throughout the rest of the growing season, particularly while the plants are in their reproductive growth stages. Those years where you expect to see high yields because you've had those timely rains throughout the season are years where you might start to see symptoms of SDS. So as I said, the pathogen lives in the soil, both in crop residue and in the soil itself. And it infects the plant through the roots. Now, infection can take place as early on as soon as that radical emerges from the seed coat. Foliar symptoms become apparent during reproductive growth stages, but if you took one of those leaflets that was showing those characteristic SDS symptoms and tried to isolate the pathogen, you wouldn't find it. The pathogen still remains below the soil surface or in the stem tissue right at the soil line. So the symptoms that you're seeing are caused by toxins that are produced by the fungus, which are carried up through the vascular tissue. They accumulate in the leaves, they're phytotoxic, and they result in those symptoms. As I said, SDS is favored by the cool, wet weather early on, and then plenty of rain during reproductive growth stages, typically between June and August. So what we have here is a table summarizing the total precipitation for each of the months, April through August, for 2010 through 2015 in Ames, Iowa. Along the bottom, you'll see what is the 30-year normal or 30-year average total precipitation for each month. Here you see in the second column whether this was considered an epidemic year or whether there was low incidence of disease. You might have noticed that those years that have above average rainfall totals in June, July, and August tend to be those epidemic years. So a researcher, Dr. Leonor Leandro at the Iowa State University, wanted to figure out just how early infection takes place and how plant age at the time of infection can affect symptom development. So what she did is she staggered planting. So she had plants that were at the seed stage or 0 days old, all the way up to 35 days old, or they had their 4th trifoliate leaf at that point. So she had plants that varied in age, and she inoculated these plants with the same concentration of the SDS fungus, Fusarium virguliforme. And then she incubated the plants for a period of days in the exact same conditions. So remember, the only thing that varied was the plant age at the time of infection. 38 days later, she destructively sampled the plants. So she pulled the plants gently out of the soil, washed them off, and then rated both root rot and foliar severity. And here's what she found as far as root rot is concerned. We have root rot severity along the y-axis and plant age at the time of inoculation in days along the x. So regardless of the time at inoculation or the age of the plant at the time of inoculation, there were root rot symptoms that developed. But what she found was those plants that were inoculated as seeds tended to have the highest degree of symptom severity. Now, she was a little bit more surprised by the foliar symptoms, And what she found was that only those plants that were inoculated as seeds showed foliar symptoms during the period of her experiment. Even those plants that were just 4 days older didn't show symptoms. Well, she was kind of surprised by these results, and so she repeated the experiment several times with very similar results. And this really just goes to show how early infection can increase symptom severity. But they wondered why they got these results. And so another researcher at Iowa State University, Dr. X.B. Yang, and a colleague, took roots from plants that had been inoculated at different growth stages, and they took cross sections of the roots. They stained the root tissue, and then they looked at the root tissue under the microscope. And what they found was that those plants that were inoculated as seeds, the fungus was able to be found in the vascular tissue in here, in those plants that were inoculated as seeds. Those plants that were inoculated as slightly older plants, the fungus wasn't able to make its way into the vascular tissue, but it hung out here in the outer stem or root tissue, in the cortex tissue, and was able to cause root rot symptoms, but not foliar disease. As you can imagine, the fungus that's in the xylem tissue, remember xylem tissue is important for water movement throughout the plant. Water that moves up and into the leaves, because the fungus is in the xylem, can result in those foliar symptoms. Okay, so we've learned a lot about symptom development, about the pathogen and those conditions that favor disease. But what about management? Let's switch gears towards management. So the first point that we'll discuss as far as management is concerned is genetic resistance. Genetic resistance has been discovered quite a while ago, and we're fortunate in that seed companies have been incorporating genetic resistance into varieties. One thing to keep in mind, though, is that there is no such thing as complete resistance. It's only partial resistance. And so in those environments that really favor disease, you'll still see some disease symptoms regardless of the level of resistance. One resource, and I have it was flowing around the room here, but I have a hard copy of this resource. It's the commercial regional trials that are conducted every year with checkoff dollars by researchers at Southern Illinois University and Iowa State University. So what happens, okay, gentleman in the back found the hard copy. He'll pass that around. So what happens every year is that seed companies will provide entries into these trials, and then these varieties will be tested in very high disease pressure environments. And depending on maturity group, they'll be planted anywhere from southern Illinois all the way up to northern Iowa. Monmouth, where I work, will be one of the locations for the 2016 trials. But what they do is they compare how much disease occurs in each of the entries to a known susceptible check for each maturity group, and then compare the two. So how they do this is at R6, or full seed, they gather and collect incidence data, so that's the percentage of symptomatic plants, and disease severity data. So this is rated on a 0 to 9 scale, where 0 is no disease symptoms, 9 is the most severe symptoms, resulting in leaf loss. And then they'll multiply these values together. So severity multiplied by incidence divided by 9 to get a number that's called the disease index, or DX for short. DX ranges from 0 to 100. And what research has shown over the years is that at R6, for every 10-unit increase in the disease index, on average, there's a 7% reduction in yield. So for a DX of 10, there's a 7% reduction in yield when compared to a non-infected crop. And so here's what the results have shown over the years. So what we have on the y-axis is the relative disease index. And this is in relation to the susceptible checks. And so the higher the number here, the more closely related all of the entries were as far as susceptibility is concerned to those susceptible checks. And so that's why I put more similar and worse up here and less similar and better down here. Because in 2005, you can see that as a percent of the susceptible checks, those entries that were included in this maturity group that year, had a pretty good level of resistance. Whereas in 2009, they were much more similar to the susceptible checks. And so what does this mean? Are seed companies making progress with resistance? Well, very likely they are. But the problem lies in the fact that we don't have a really high level of disease each year. And so those years where we have low levels of disease, you can imagine that the breeders who are, soybean varieties move in and out of the market so quickly, the breeders don't have a lot of years to evaluate and make selections for disease resistance. So those years where there's low levels of disease, they're not as likely to make good resistance selections. So researchers and farmers have suspected that there's a relationship, some sort of relationship between the soybean cyst nematode and SDS. And so there's a series of greenhouse studies at Iowa State University done by Darren Mueller and the nematologists there at Iowa State University, where they looked at symptoms and when those symptoms developed in plants that were infected with the fungus alone, the nematode alone, or the combination of all three, both pathogens on the soybean plants. And what they found is that symptoms of SDS may appear earlier and can be more severe with SCN infection. It's also thought that the pathogens may spread together. They can spread in the same way with tillage implements, let's say. Anything that brings soil from one field to another. And so this is why current management recommendations are to select varieties with high levels of resistance to both SCN and SDS. Let's switch gears to talk about chemical control. So what we know about the pathogen and when it infects, and when and where and what time in the growing season that we typically apply foliar fungicides, we know that there's very little chance that foliar fungicides will come into contact with the pathogen at the time of infection. And this is why seed treatment fungicides were more likely to be of use when it came to SDS management. And so one of Dr. Carl Bradley's students, before 2015, because this is when the paper came out, one of Dr. Bradley's students looked at 11 different commercially available fungicidal seed treatments. And he compared them to an untreated control in various field trials and greenhouse trials over many years, locations, and different varieties as well. So in addition to looking at symptoms, he also dug up roots at various times throughout the growing season and extracted and quantified pathogen DNA from the roots to see just how much of the pathogen was in the roots. And what he found was that some of the seed treatments actually reduced the amount of pathogen DNA in the roots, but this didn't translate to any effect on symptoms. And so what this meant was that although some of the seed treatments might have had an effect on the pathogen itself, enough of the pathogen was able to make its way into the roots to still cause symptoms. More recently, in 2013 through 2015, there were a couple more seed treatments that were developed and specifically marketed towards SDS management. There was a series of uniform seed treatment trials that were conducted in 10 different US states and in Ontario, Canada. And they looked at 3 different products. So Alivo, which is a product from Bayer Crop Science. The active ingredient here is fluopyram. There's Myrtect. The active ingredient here is thiabendazole. This is a product from Syngenta. And then there's a product that might not be as familiar to folks, and this is a product called Heads Up. This is a biological seed treatment, and it's a biological extract. It's actually approved for organic production, and it's produced by a company called Heads Up Plant Protectants Incorporated out of Canada. And so they looked at these 3 seed treatments over multiple locations and multiple years. And here's a product comparison over 7 different locations. Each of these columns are the results comparing a base seed treatment with the SDS treatment and the base seed treatment without the SDS treatment. So Poncho Votivo and Alivo are over here. Myrtect is here, Heads Up is here. The black bar is the reduction in SDS disease index, so the percent reduction. And the red bars is the yield response. You'll see that Alivo reduced SDS by a little bit more than 5%. And when compared to that base seed treatment, Alivo also resulted in a yield response that was more than 3 bushels per acre. So they decided to look into the Alivo results a little bit further. And so here's the data broken out by each year. The black bars is 2013, red 2014, and yellow 2015. The columns on the left are total disease index. Columns in the middle is SDS reduction. And then columns on the right is the yield response. And if you farm anywhere near Western Illinois, so I'm in Monmouth, you'll know that 2014 and 2015 were pretty good years as far as disease was concerned. We had high levels of disease in both years. And they did in their trials as well in 2014 and 2015. You can see that the results as far as reduction of disease and yield responses were greater in those years where there was higher disease pressure. Here's how different levels of genetic resistance and the Alivo seed treatment panned out. So here are the different number of observations for both susceptible and moderately resistant varieties. You can see that there was a significant amount of disease in both the susceptible and the moderately resistant varieties. And Alivo was able to reduce symptoms in both the susceptible and the moderately resistant variety. But again, disease pressure was higher in that group of susceptible varieties, and so the yield response was a little bit higher there as well. Here's the Alivo results broken out by planting date. So before May 1, first half of May, second half of May, and then after June 1. You can see in these trials, the May planting dates had the highest disease indexes, and that correspondingly, Alivo had the highest level of symptom reduction and the highest yield response under the highest disease pressure. Here's the results of Olivo broken out by final disease index. So 0, no disease, all the way up to more than a DX of more than 20. And you can see the average for each of these categories right here. So this high level of disease, this was about halfway to the worst disease you can get. You can see that where there was no disease, there was a slight yield response with the Olivo, but it was under the higher disease pressure where you had higher yield responses for Olivo. But even Olivo has its limits. So really, really high levels of disease, you still will get a yield response, but it's not magic. So soybean seedling injury is a common occurrence, and it's sometimes very difficult to figure out what the causes of seedling injury are. This might be something you see in your own fields. What we have here is Pythium blight. Here's a injury caused by pre-emergence herbicides. And here is injury that's caused by Olivo. So this is termed a halo effect because it kind of rings the outside edge of those cotyledons. And so research was conducted in 2014 and 2015 to see whether some of these pre-emergence herbicides and Olivo injury itself, whether that Olivo injury increased with the pre-emergence herbicides, whether there was any kind of interaction. So in Indiana and Iowa, they looked at susceptible and resistant varieties. Again, that base seed treatment and base plus Olivo. And then they tested, using these variables, several common pre-emergence herbicides. Within each plot, they looked at a lot of different things. So they looked at phytotoxicity symptoms very early on and a little bit later on. They did stand counts. They looked at disease index and then yield later on. What they found is that damage is visible early on, but it typically goes away by that 4th trifoliate leaf stage. They also found that the symptoms of phytotoxicity can increase with certain pre-emergence herbicides and cool, wet conditions, but that there was no negative effect on yield. So this is a picture I took in Monmouth this past year. You can see that halo symptom right here. I can tell you that in Monmouth just last year, the plants were indistinguishable from those that had no seed treatment after that 2nd trifoliate leaf. And that was in Monmouth last year. Okay. So here's a summary of the seed treatment results. So the Olivo can reduce SDS when it's present. It can also result in a significant yield response, but these yield responses are higher under higher disease pressure. Earlier planting dates saw a higher yield response dropping off in June, and then when disease indexes were higher, there was the best response. So let's talk now about cultural practices, and we'll visit about crop rotation, tillage, and planting date. A survey by the Iowa Soybean Association in 2014 showed that 96% of Iowa soybean farmers used crop rotation to manage sudden death syndrome. Now, the primary rotation crop in Iowa is corn, pretty similar to our, at least in the northern half of Illinois, our crop rotation as well. What research at the Iowa State University showed was that the fungus that causes sudden death syndrome can be isolated from the soil itself, from soybean residue, roots, seeds, and from corn residue, roots, and kernels. And so the typical corn and soybean rotation won't do a whole lot as far as inoculum reduction. So corn and soybean rotation, rotating the corn, won't necessarily reduce the amount of inoculum. So to look at a slightly longer rotation, researchers looked at a corn and soybean rotation compared to other rotations. 3-year rotation, which is corn and soybean, with oats and red clover in that 3rd year. 4-year rotation, corn, soybean, oats, and alfalfa, and then alfalfa. So 2, 3, and 4-year rotations. And what they found, so here's the disease index. From left to right, we have 2-year, 3-year, 4-year rotation, is that the longer term crop rotations at this location saw a lot less disease symptoms and incidence over these high disease years. It's much easier to see in 2010 and 2013.
Would that be the same if it was corn rotation? Since the inoculum or the disease survives on corn residue?
So that's a really good question. So the question was, would these be similar results if we had multiple years of corn and then grew soybeans? And we haven't done that experiment yet. And I know that many people in Illinois do the corn-soybean rotation. And so that's something to look into. And I'm not sure if corn or soybeans are a better host of this pathogen, but I would suspect that there wouldn't be much of an effect just because both of them are hosts of the pathogen. And so it's not like we're going to be reducing inoculum. Another question?
Does research with cover crops?
You read my mind. The question was regarding cover crops. That's going to be the next slide. Okay. So this is what this looked like. So here's the 3-year rotation, and here's the 2-year rotation. Here's the cover crop slide right here. So researchers at the University of Minnesota and Iowa State University looked at other crop species to see whether they were hosts and could cause root rot symptoms if they were inoculated with Fusarium virguliforme. So they inoculated the plants, and then they rated root rot symptoms. And what they found is here. So we have soybeans over here, alfalfa, crimson clover, hairy vetch, pea, red clover. So what they found was that other plants can be hosts of this fungus, specifically a lot of the other legumes, so alfalfa, crimson clover, red clover, pea, and sorghum. They looked at root rot, so this pathogen significantly increased root rot severity. I'm not sure that they looked at pathogen reproduction here though. You will notice that sorghum is also a host. But if you look here, here's mustard, turnip, oats, ryegrass, and rye, some of our more common cover crops. The pathogen wasn't able to cause root rot on these species, and so I'm pretty sure that those would not be a host of the pathogen. Okay. So there was also research looking at soil drainage, compaction, and tillage. I know that in 2015 at the research center where I worked, where we typically saw symptoms develop first was in our most compacted soils. So there's some evidence that improving soil drainage and correcting compaction may reduce the risk for disease. Reports on tillage are very inconsistent. Some of the research shows that certain tillage techniques can reduce disease, others show that there's no effect. One thing that we're pretty certain about is similar to the soybean cyst nematode, we're pretty sure that tillage and tillage implements can move this pathogen, the soil-borne pathogen, around within fields and then between and among fields as well. So to kind of get a handle on tillage, a study was established after 2010 in Iowa. So here's 3 years worth of data in a corn and soybean rotation. The different tillage treatments are here, from the least amount of tillage, so no till in both crops, to no till before corn, chisel plow before soybeans, disking before corn, chisel plow before soybeans. And what they found as far as plant stands, disease index, and yield were concerned, they found no significant differences for the different tillage treatments in a corn and soybean rotation. And that may be a function of the fact that both of these are hosts of the pathogen. Here's our last point. So a series of planting date trials were established in the 3 I states in Ontario in 2013 and 14. They targeted the end of April, middle of May, end of May, and the middle of June as the planting dates. Here's the disease severity results. They averaged across cultivars in all the locations, and here's the different planting dates on the bottom. The lighter blue bars are root rot severity. You can see that root rot tended to be highest in the earliest planting dates, falling off with the later planting dates. Foliar severity, though, was highest in the early June planting dates, was very high also in early May and late May, falling off in mid-June. This really points to the fact that these foliar symptoms are very dependent on those timely rain events during the potting growth stages. But keep this picture in your mind right here, how there's more symptoms early on, tending to fall off in mid-June, because I'm going to show you the yield results next. Here's the yields. What they saw here is that the earliest planting dates resulted in the highest yields, regardless of disease severity, for both roots and foliar symptoms. This is because the earlier that you plant, timely planting, not too early, the earlier that you plant, the longer growing season that soybean crop has. So it's able to more fully reach its yield potential. Don't get me wrong, I would imagine that yields would be much higher without those symptoms that they saw. But it's still the early May planting date resulted in the highest yields. And so to summarize, crop rotation away from corn and soybeans might be an option, probably not an economically sound option for many people. Note that some of the legume crops, some of the cover crop species may be hosts. Tillage in that study had minimal effect on SDS. And planting date, root rot symptoms increased with earlier planting. Foliar symptoms really depend on those summer rains. And it's probably not a good idea to adjust planting date solely to manage sudden death syndrome. And so to summarize here, genetic resistance, make sure that you select varieties. You've probably already made your variety selections, but it's important to select varieties that have a high level of resistance to both sudden death syndrome and SCN. We now have a new tool as far as chemical control is concerned, and your plan for how to best manage sudden death syndrome will have to depend on the history of that disease in your particular field and your comfort level, your level of risk that you are personally willing to take. Thank you for your attention. Here's my name, where I work, my email address, phone number. I do have cards up here. I was told probably about 10 minutes ago I had 5 minutes. It sounds like other people are still going though, so I'd be happy to take questions if you have any. Questions? Yes, ma'am.
We had a field in eastern Iowa that was 9 years of continuous corn, and it's still, they came in and the SDS still took out the beans in the 10th year. And so that must be why, because it's still growing some of their crop residue.
Yes, so the comment wasn't necessarily a question. The comment was this young woman planted corn for nine continuous years and then planted soybeans and got nailed by SDS. And she commented that it's probably because corn is also a host for this pathogen, and I think you're exactly right.
So you think it would take 1 year of like a completely different crop, or since it was built up and lasted that long, that it might take more than 1 year?
It might take more than 1 year. I'm not sure, because I don't know that they looked at the level of inoculum in the soil in those longer term crop rotation studies, but I would imagine that it might take some time.
Did you mention anything about cleaning tillage equipment? Like as far as, is it transferred in the soil? I mean, that's,
Sure.
I mean, would that be, now that it's already, you know, in a lot of fields, would that be even worth doing? Like, can you make it worse
Sure.
than it already is not cleaning them, I guess?
So the question is about sanitization of tillage equipment and whether it would be worthwhile between different fields. I would say in some of those areas where we haven't seen disease yet, it probably would be a good idea, but I would imagine that people have probably already spread it from field to field not knowing. And because, you know, disease is so dependent upon environment, we might have that pathogen and not even know it. So it wouldn't be a bad idea, just it might take some time. I don't know that you would get all the soil off of your tillage equipment anyway.
Yeah.
But it's a good thought. Okay, I've been told that the giant hook is coming out and I'm ready to go. All right, so thank you.